So, welcome back to this last part of this lecture about the statistical approaches to variant and genotype calling. Before we start with that, just one brief word about the different pieces of software that have been written to do this task. So there's a whole slew of different variant genotype callers available today. And so when looking at what to pick here in terms of representative approaches, I really had a choice of many things, and I just decided for three rather typical approaches that contrast very well, so that I can show you the advantages, disadvantages of it. Name one piece of representative software each that implements that approach, and everything else is up to you for later, either further study if you want to, or later if you're really doing bioinformatics. So it's just a selection of three prototypical approaches out of many that are around. So with that, let's look at those three. Of course, we start with the simplest thing, the obvious way, count and classify again. So this approach is basically just an extension of what we've done before for just variant calling. So now we're dealing with a diploid organism, let's say. So instead of just doing a majority vote whether there is a variant or not, as before, what we do now is at every site with at least one non-reference base count, we count the bases supporting the reference supporting and the non-reference bases. That's like before, but instead of now looking whether more than 50% of bases are variant, we do different, more complex thresholds. So we could, for example, go with a 20% threshold first, and then if the non-reference bases constitute less than that threshold of, say, 20%, then we call a homozygous reference genotype. In other words, we say there is no variant at the site. If it's intermediate, let's say the non-reference base constitutes more than those 20%, but also less than another second threshold of 80%, then we call a heterozygous genotype, meaning we assume that this intermediate finding is because one of the two copies in the diploid genome carries the allele, the variant allele, the other one doesn't. And then finally, if the non-reference bases constitute more than this threshold of 80% of all reads, that's when we decide to call a homozygous variant genotype. And with this very simple adjustment, we have a decent genotype caller available, right? It, with just like its simple predecessor, our simple first variant call, its main advantages: it's super easy to understand, also super easy to implement, and it will be fast because it's just comparing read counts against thresholds, and that's really not computationally expensive at all. There are disadvantages too, of course. So first of all, we discussed the sampling problem before, and what we're doing here is not solving this problem, because of course the ratios of variant alleles versus reference alleles that we're relying on in this thresholding approach, those ratios become very unreliable at low coverage sites. Ratios change quickly if the numbers that give those ratios are small. And of course, in this approach, we always treat multiple samples independently. So this might come as a bit of surprise, but we'll see later that there are better algorithms that actually can do information borrowing between multiple samples in the same analysis to overcome low situation at low coverage sites, at least partially. Finally, also, our simple algorithm does not take read mapping and base qualities into account, but we know from our first example that this is easy to fix. So just like there, we could now again weigh our observations with the mapping quality, with the mapping error probabilities and base error probabilities, and then use those weight scores to incorporate these measures into our final decision. So that would be rather straightforward to do. And overall, such variant callers are not bad as long as you have decent coverage, and of course if you're not dealing with too high ploidy. So they work well for the diploid case, but of course you have to come up with a different set of thresholds for every different ploidy. So this 20 and 80% might work well for the diploid case, but it would be complete nonsense for the triploid case, for example. Still, those variant calling tools like that are in widespread use. Representative software would be SAMtools MPileup combined with custom scripts. So SAMtools is a tool suite made for manipulating SAM-BAM data, so the typical input to variant calling tools, and the MPileup tool within that suite is good for producing, as the name implies, those pileups of reads from the BAM file, and then you just basically go through the pileup that you're literally then obtaining as a tabular file, where each row in the file is a read position on the genome, and then you have all the bases tabulated that provide information for this position, and then you pass that file with custom scripts to do the actual variant calling. And that's not hard to do, and it's quite successful already. However, you could do better, and if you're willing to work a bit harder to get the implementation right. And so one thing you could do, you could treat this as a pure classical statistical problem. So classical statisticians, they like the null hypothesis, and they also use it here. So in this case, your null hypothesis would be there is no variant at that site I'm looking at. Right? So you go through the sites one by one in the pileup. Your null hypothesis at every site is always there is no variant, and then you're trying to prove the opposite or get evidence for the opposite. And reject that null hypothesis. So what you would do is you would calculate the probability that the number of variant supporting bases, or an even higher one, that you're observing at that site in your actual data are simply due to base calling errors. And for this, you can use the so-called Poisson binomial distribution. It has this complex formula. It's well-known distribution. You can look it up in Wikipedia if you don't know it yet. And so you would what this formula takes into account is the error probabilities of each reference supporting base and the error probabilities of each variant supporting bases, puts it into that formula, and gets an overall probability that the variant supporting bases observed are just explainable with these errors alone. If you were to take a bit more effort, you could incorporate also the mapping qualities into that formula. It would get more complex, even more calculation, but it's certainly doable. And then if the added probabilities of all that if the added probability is that the observed number of variance supporting bases and any number higher than that combined are less still than a fixed p-value, then you conclude, okay, I cannot explain the number of observed variance supporting bases by these error ratios alone, so there is likely a variant there. And then you reject your null hypothesis and call the variant. Well, clearly, advantage here takes base quality into account by default. It's easy to modify, though the formula gets more complex, to take also mapping quality into account, and it's the proper statistical treatment of the situation. It's more accurate, statistically more sound than just counting numbers of bases and using arbitrary thresholds. But now you have a solid statistical basis for your decision making, and you're not just making up thresholds that seem to fit. There are disadvantages too. These statistics are rather expensive to compute, and especially at sites with high coverage. So if your coverage for some reason is very unequally distributed across your genome, and you have some sites that are really, really high coverage sites, then your software might run for a really long time on such a data set. The other drawback here is that there is no explicit handling of genotypes anywhere, but only of variance. So you only reject the null hypothesis, there is no variant, and you find there is a variant instead. But then the genotyping itself for called variance has to be done in a follow-up process somehow, possibly through thresholding again. And again, it treats multiple samples independently of each other. Again, no information borrowing like with our simple approach. Representative software for this kind of variant callers is Loafrec. There's a GitHub repo that you might check out. It's might want to check out. It's C++ source code, relatively easy to read. Most of it is implementing those statistical formula in code, and it's considered a very accurate and sensitive variant caller, a bit slow running at high coverage sites, and it doesn't call genotypes. It can only tell you variants, but not make any claims about genotypes. So something, I mean, even our simple, obvious way had genotypes included, right? But the more advanced classical statistical way doesn't. So is there a more sophisticated approach that can treat genotypes? Yes, there is, and there's a really good one, and that's the Bayesian way. So Bayesian variant callers, of course, rely on Bayes' theorem, remodeled to work with genotypes and sequenced reads. And so the formula we're using here is that simple equation, well-known Bayes' theorem, that the probability of a genotype, of any genotype, given a set of observed reads, so that's the S in the formula, equals the probability of observing this set when the genotype is actually genotype G times the probability of the genotype alone divided by the probability of the set of observing the set of reads, just like that, unconditionally. So, wow, that's a hard to pass sentence, but it's actually not so bad as it first looks. So your goal here is to find the genotype G that has the highest probability given your observed set of reads. So that's trivial goal, of course, that's what you want. There's one crucial simplification that we can do right away in such an algorithm. and that is that the probability, the unconditional probability of observing the set of reads we observe is of course always small, because there's nearly endless possibilities what kinds of reads we could observe, and it's also unknown. But what we know is that it's constant across genotypes, because it's not conditional on the genotype. So that denominator here is a constant, and if it's a constant and we're only interested in finding the maximum probability given the set of reads, and that is a constant, then we don't need it. Now we only need then to find the maximal value of the numerator, and we have what we want. So we can kick out this unknown probability of the set of reads immediately and simplify our formula to just the simple part here, or simpler part. Now let's tackle the remaining parts one by one. So how would you calculate the probability of observing the set of reads given a certain genotype? Well, first step, you assume a genotype. There aren't that many possible ones, right? If you have a diploid organism, your genotype can be either homozygous reference, heterozygous, or homozygous variant. Now you just go through iterate through those three possibilities, and you assume each of those genotypes in turn. So let's start, for example, with homozygous reference. Then what you know is that any given base B you're observing in the set of reads could represent one allele, let's call it A, of the genotype. So the probability of the base being the result of the genotype, a diploid genotype consisting of allele 1 and allele 2, A1 and A2, is simply the average of the two probabilities of observing the base given one of those alleles. The average, because you add them and you divide by 2, and this is the probability of observing the base when you have a certain diploid genotype. Now then you multiply all those probabilities for all bases in this pileup column, and that's how you obtain the probability of observing the set of reads giving the genotype. Because everything left and right doesn't really matter, because it will be independent of the genotype, and then again you can neglect it. So you only have to look at this one pileup column with that one column of bases, and only have to calculate these probabilities of those bases given the genotype. And then you repeat this for the other possible genotypes, right? And then you have an array of probabilities conditional on each of those genotypes. And then lastly, you calculate the probability of the genotype unconditionally. And there are two ways to do this. So if you have only one sample or a very limited number of samples in your analysis, you simply assume a constant average mutational load across the genome. So from evolutionary theories and observations, you can come up with a measure of mutability of genomes, and let's say you assume that your genome acquires on average in every individual one mutation per 1,000 bases. You would have a mutational load value Q of 0.001, and then that's the likelihood of having a mutation per genome. Now in a diploid genome, of course, that chance is twice as high. So the probability for a heterozygous genotype is then 2 times that mutational load, so it's 0.002 in our case. And you can then calculate the probability for the homozygous reference genotype and the homozygous variant genotype as these values. And you do this with a formula named after these scientists, Hardy-Weinberg, the Hardy-Weinberg equilibrium, which states that the probability of the homozygous reference genotype is 1 minus this mutational load squared. As I said, the heterozygous probability is 2 times it's like a binomic formula. So Q squared is the probability of the homozygous variant genotype, and this is the 2 times the product of the outer two. So 2 times 1 minus the mutational load times the mutational load. And then if you fill in the Q 0.01, that's how you derive these values here. So the other way, the more interesting way of then assuming a constant average mutational load is actually estimating the genotype likelihoods from the samples themselves. So basically you're doing a first round of unbiased genotype calling, and then from the genotypes you estimate the frequencies with which they occur. This doesn't make much sense for 1 or 2 or 3 samples, but if you have sampled a population of, say, 1,000 individuals of your organism, you have sequencing data for these 1,000 individuals from one species, you can actually look at each variant site and say, why, is this a rare variant or is it one that occurs very frequently in this population? And this will affect your prior in Bayesian terms that you give to a certain genotype. Yeah? If you know this is an incredibly frequent variant in this population of individuals, and you have an individual with a borderline variant reads supporting pattern. So let's say you find out of 10 reads, you find 2 that support the variant, and you're in doubt whether that's just sequencing errors or whether it's really a heterozygous genotype there. If you know that the genotype is, that the variant is quite frequent in that population, the chance increases knowing that this individual is actually heterozygous, right? If it's a very rare variant, on the other hand, then you're not observing in any of the other 999 individuals, then you would rather argue that probably this one individual then has a sequencing error in there and is actually homozygous reference, right? This is roughly how that works. So you can bias these genotype frequencies that you calculated here, the a priori assumptions, with the data you actually observe. And now what you're doing is you combine the two. So now you have this probability of every genotype, and you have the probability of observing the set of reads, the actual observation, conditional on each genotype, and now you're just seeing which combination gives the largest product. So Bayesian stats is really tough to explain. I'm sorry for that in words, but it's actually quite logical if you think of it. Yeah, you're not just doing this is kind of the classical part. You observe the reads and see what evidence do I have. But in Bayesian stats, you're also taking this prior into account, this prior knowledge, how likely each of the genotypes actually is. So even if a certain set of reads seems to favor a certain genotype slightly. If you know that genotype is very, very unlikely, you will not call that genotype, but the slightly less supported but more generally likely genotype instead. Right? Think of a very rare variant. The chance to see this in any individual in a homozygous state, homozygous genotype, is very, very low, because it's the likelihood of the mutation as such, of the variant as such, squared. So it's very, very low. So if you have, even if you have sequencing data, read pileups that seem to confirm that homozygous genotype pretty well, so the probability of observing your set of reads, assuming homozygous genotype, is considerably higher than the probability of observing the same set when the genotype is heterozygous, you would still think it's, for a long time, that it might be in fact a heterozygous genotype, simply because the heterozygous genotype is so much more likely for that very rare variant than the homozygous one. So I hope that's kind of understandable. It's hard to explain in human language. That's where mathematics really have their appeal. Anyway, this kind of complicated but very logical algorithm has been implemented in representative software called Freebase, a really interesting, cool variant caller, which uses that approach, and it's, as you can imagine, particularly powerful when you have a priori knowledge about your genotypes, or you have large sample sizes and can borrow information from these samples, and then it helps you get good genotype calls even at relatively low coverage sites. So that was the comparison of the three approaches, and now there's only some minor things left. So one thing is how, now assuming we have a variant caller that also calls genotypes, so a genotype aware variant caller, what does it output? Well, it also outputs variant calling format VCF, like any standard variant caller, but such genotype aware callers are free to define additional genotype columns in that the VCF specification allows for. So the minimal set of columns that a VCF has to comprise are the ones we already know about, those first 8 columns. But there can be additional ones, and those are then the genotype columns. And so if you want to have genotype columns, what you need is first a format column that defines how the following genotype columns are structured. There's a couple of keys that are standard, like GT, genotype, GQ, genotype quality, and so on. And so those genotype call statistics are always sample specific. So if you have multiple samples in your analysis, you would have multiple columns, one for each of your samples, following the format column. And then each of those sample columns would show the overall structure that the format column specifies, meaning each of those columns in this case would start with a genotype code, separated with a colon from the next value, which would be the genotype quality, which is again this log scaled thing, the inverse of the error probability, so the probability that the genotype caller might have called the wrong genotype, and the higher the value, the less likely an error in the genotype calling is. And you would have this for every sample in that analysis. That's the extended VCF format. And yeah, the only other thing we have to discuss is what you then actually do with those variant lists that you produce. So how do you interpret them? And this is a biological question which is very, very important and also already should be thought about before actually performing the sequencing experiment, because proper study design is quite important here. Because fact is that almost all organisms always contain some variants in their genomes. That's because evolution happens. Copying of genetic information is nearly perfect, but only nearly so, because if it was perfect, then life on Earth would stop evolving. So the exact same DNA sequences would be passed on to all progeny of all life forms eternally, and there would be no room for evolution anymore. So that's why a little bit of error always happens during replication. And now, because genomes are really, really huge typically, as we've learned, thousands to hundreds of millions of bases in size, there are always several variants in each genome. And so, for example, we humans, when you compare two human beings to each other, even though they will share like 99.9% of their genomic information, that still leaves enough variance in this 0. Something percent that each of us carries thousands of SNPs that distinguish us from the human reference genome, right? And not all of them are really in sequences that code a protein. Some are in introns, in intergenic regions between genes, but still hundreds are actually encoding sequences and could influence our phenotypes, so our genetic traits that we develop. And so those differences is what makes us different as individuals. Doesn't mean that those variants make you sick, they just make you different. So if you're looking for variants of clinical relevance or of biological significance with respect to one specific biological question, it's not enough to just build a variant list, because this will contain many things and not just the one thing you're looking for. So you would end up with a variant list of thousands of variants, and then you would not be much wiser than before conducting the experiment. What's instead really important is that you always do variant calling for multiple samples, so that you can then compare the variance between those samples. And you should typically choose samples in a way that some of the samples show the phenotype you're interested in, for which you suspect that a certain variant must be responsible, and other samples that don't have the phenotype. And then what you would do is kind of variant subtraction. So you would look, okay, is there anything shared by my phenotypic samples, any variant that is not present in any of the non-phenotypic samples? And this is how you reduce your variant list normally dramatically. That's why, for example, in clinical settings, you're studying, for example, family trios. So if you have a child with a genetic disease, you would also sequence the genome of the father and the mother, and possibly other family members, and then you conclude, you look at the variant patterns you're finding, and then you're asking, is there anything in the genomes of the individuals affected by the disease that is not present in the related individuals that are not affected by the disease? And then you can narrow down your variant list. In addition, variant annotation is pretty important, so you can predict from genetic code and gene architecture that we all discussed in initial lectures, what the effect of certain mutations would be on known genes and proteins. And if you know something about the protein function, this might help you predict what the effect of the variant might be. And then if you study a genetic disease and you find a variant that affects a protein that is known to play a role in that disease process, then of course you would prioritize that variant for follow-up experiments much more than a variant, say, in an intron of a not annotated gene you do not know anything about, right? So this is another way to get a lower number of candidate variants. So the simple approach that you just take one sample, you sequence it, you do read mapping, and you do variant calling then, and then you have your causative variant for what you're interested in. This is really only working in rare cases, like the simple ones, like the motivating example with the bacteria. This might work. You would end up with maybe 5 candidate variants. One of them is the one you're interested in. But in any more complex setting, this is not a very promising approach. This is actually something I want to highlight in the exercises, which will this time be rather biological, so it takes a bit of detective attitude, so you have to do a bit of careful thinking, rule out possibilities. I'm actually curious to see how you're coping with those exercises, because they are relatively off topic compared to what we talked about now algorithmically, but it could have you understand this last aspect that I was focusing on now with the variant interpretation actually a bit. So curious to see how that goes. Okay, and with that I'm at the end of this lecture for today. I'm also done with my part of this lecture series. I hope you enjoyed it, and I hope you will enjoy the rest of the series still. And then, yeah, I already wish you good luck for the exams, and yeah, have fun. Bye bye.